This is Week 8, Genetic Variation in Populations, Part 1. The learning objectives for this week are to give examples of genetic disorders occurring at high frequency in particular populations, define the concepts of genetic drift and founder effect, use the Hardy-Weinberg distribution to calculate the relationship between gene frequencies and phenotype frequencies, and to determine genetic risk to individuals, to describe ancestry testing, and to discuss the use of linkage association studies and genome sequencing in identifying disease genes. The language of genetics at the family and individual levels is written in DNA sequences. However, at the population level, the language of genetics is allele frequencies. Genetics at the population level goes beyond science, embracing information from history, anthropology, human behavior, and sociology. Population genetics enables us to trace our beginnings, understand our diversity today, and imagine the future. Allele frequencies that do not change in response to environmental factors provide a way to distinguish individuals. We will now look at some definitions that you all need to know. First one is population. It's any group of members of the same species in a given geographical area that can mate and produce fertile offspring. Population genetics is a branch of genetics that considers all the alleles of all the genes in a biological population, which constitute the gene pool. The gene pool refers to the collection of gametes in the population. An offspring represents two gametes from the pool. And gene flow. Alleles can move between populations when individuals migrate and mate, which underlies evolution. In this slide, we see that allele frequency is equal to the number of a particular allele over the total of a number of alleles in the population. You count both chromosomes of each individual, and the allele frequencies affect the genotype frequencies, which is the frequency of two homozygotes and the heterozygote in the population. Phenotype frequencies are determined empirically by observing how common a condition or trait is in a population. Genetic counselors use phenotype frequencies to estimate the risk that a particular inherited disease will occur in an individual when there is no family history of the disease or illness. If we look at this side on phenotype frequencies, we can look at PKU, which we studied previously, and we can look at it in various populations. As you can see here, the incidence of PKU in the Turkish population is far greater than any other group that is listed here. Shifting allele frequencies in populations reflect small steps of genetic change. These are called microevolution. These small incremental changes alter the genotype frequencies and underlie evolution. Genotype frequencies rarely stay constant. They can change under any of the following conditions. Individuals of one genotype are more likely to reproduce with each other than with individuals of other genotypes, which is non-random mating. Individuals move between populations, migration. Random sampling of gametes alters allele frequencies, genetic drift. New alleles arise, mutation. People with a particular genotype are more likely to produce viable, fertile offspring under a specific environmental condition than individuals with other genotypes. This is natural selection. All conditions that were just discussed are common with the exception of mutation. Therefore, genetic equilibrium is rare. Remember that this is when allele frequencies are not changing. Microevolution is almost unavoidable given our practice of picking our own partners and of moving around in our society. When sufficient microevolutionary changes accumulate, then we have what is called speciation, the formation of a new species. These changes can occur through many small changes over time and and/or a few changes that greatly affect the phenotype. In 1908, a British mathematician, Hardy, and a German physician, Weinberg, independently used algebra to explain how allele frequencies can be used to predict phenotypic and genotypic frequencies in populations of diploid, sexually reproducing organisms. They both disproved the assumption that dominant traits would become more common, while recessive traits would become rarer. Please visit the links to two YouTube videos on both Hardy and Weinberg, as well as the Hardy-Weinberg equilibrium. The binomial expansion used to describe genes in populations became known as the Hardy-Weinberg equation. It can reveal the changes in allele frequency that underlie evolution. If the proportion of genotypes remains the same from generation to generation, as the equation indicates, then that gene is not evolving or changing. This situation, Hardy-Weinberg equilibrium, is theoretical. It happens only if the population is large and undisturbed. That is, its members mate at random, do not migrate, there's no genetic drift, mutation, or natural selection, as we discussed earlier. Please look at this equation. P is equal to the allele frequency of one allele. Q is equal to the allele frequency of the second allele. P plus Q equals 1. All of the allele frequencies together equals 1. P squared plus 2PQ plus Q squared equals 1. All of the genotype frequencies together equals 1. P squared plus Q squared is the frequencies for each homozygote, and 2PQ is the frequency for heterozygotes. This slide is showing a variation on the Punnett square, and it reveals how the random mating in the population between two heterozygotes can give you the genotypes of homozygous recessive, homozygous dominant, and heterozygote in the relationship of P squared plus 2PQ plus Q squared. Let's look at an autosomal recessive trait to solve a problem using the Hardy-Weinberg equation. This trait is a middle finger that is shorter than the second and fourth fingers. If we know the frequencies of the dominant and recessive alleles, then we can calculate the frequencies of the genotypes and phenotypes and trace the trait through the next generation. The dominant allele, uppercase D, confers normal length fingers. The recessive allele, lowercase d, confers a short middle finger. We can deduce the frequencies of the dominant and recessive alleles by observing the frequency of homozygous recessives, because this phenotype, short finger, reflects only one genotype. If 9 out of 100 individuals in a population have short fingers, genotype DD, homozygous recessive, the frequency is 9 out of 100, or 0.09. Since little d homozygous recessive D equals Q squared, then Q equals 0.3. Since P plus Q equals 1, knowing that Q is 0.3 tells us that P is equal to 0.7. And next, we can calculate the proportions of the three genotypes that arise when the gametes combine at random. As you can see on this slide right here, we can figure out what this would be. So for the homozygous dominant, it would be 0.49, 49% of the individuals in generation 1. For the homozygous recessive, it would be 9% of individuals in generation 1. And for the heterozygous individual, it would be 42% of the individuals in generation 1, as one can see from this slide. If we now jump ahead a few generations and assume that people choose mates irrespective of their finger length, this means that each genotype of a female is equally likely to mate with each type of a male. Then we can look at this table. And in this table, it multiplies the genotype frequency for each possible mating, which leads to offspring in the familiar proportions of 49% homozygous dominant, 42%, 9% homozygous recessive. This gene, therefore, is in Hardy-Weinberg equilibrium. The allele and the genotype frequencies do not change from one generation to the next. To derive carrier risks, the Hardy-Weinberg equation is applied to population statistics on genetic disease incidence. To determine allele frequencies for autosomal recessively inherited characteristics, we need to know the frequency of one genotype in the population. This is typically the homozygous recessive class because its phenotype indicates its genotype. I would like to talk a little bit about cystic fibrosis. In history, we have known of this disease for centuries, and it has been written in history that woe to the child who tastes salty from a kiss on the brow, for he is cursed and soon must die. Cystic fibrosis is an autosomal recessive disease that was first diagnosed in 1938. It is the most common autosomal recessive disease in white children, and mutations in the CFTR gene can account for this, and there are currently over 1,900 mutations that have been found. The most common mutation is F508 deletion, which accounts for approximately 70% of the CFTR genes in patients with CF worldwide. If we look at how this gene was discovered, we see that Dorothy Hansen Anderson discovered the disease, and she wrote of cystic fibrosis in the American Journal of Diseases of Children in an article called Cystic Fibrosis of the Pancreas and Its Relation to Celiac Disease, a Clinical Pathological Study. And the year was 1938. If we look at the pathophysiology associated with cystic fibrosis, we see that the mutations in the CTFR gene affect the ion channels that chloride movement out of the cells utilize in certain organs. If we look at the lungs, a thick, sticky mucus can accumulate and create an environment that's very hospitable to certain bacteria, such as the opportunistic pathogen Pseudomonas aeruginosa. We do not find infections from such organisms to be common in healthy lungs. In the pancreas, a clogged pancreas prevents the pancreatic digestive enzymes from being released to go to the duodenum for digestion. Thus, this impairs nutrient digestion. We also see infertility problems in both males and females with these mutations. The incidence of an autosomal recessive disease in a population is used to help calculate the risk that a particular person is a heterozygote. Returning to the example of cystic fibrosis, the incidence of the disease, and therefore also of carriers, may vary greatly to different populations. Please look at this chart that shows African Americans, Asian Americans, Caucasians of European descent, and Hispanic Americans. If we look at this, we can see that Caucasians of European descent are far more likely to be carriers for the cystic fibrosis genes. And if we look at Asian Americans, they're only 1 in 150. CF affects 1 in 2,000 Caucasian newborns. Therefore, the homozygous recessive frequency, lowercase c, if lowercase c represents the disease-causing allele, is 1 over 2,000 or 0.0005 in the population. This equals q squared. The square root of q squared is about 0.022, which equals the frequency of the lowercase c allele, the homozygous allele. If q equals 0.022, then p, or 1 minus q, equals 0.978. Carrier frequency is equal to 2pq. which equals 2 times 0.978 times 0.022, which equals 0.043, which is equal to 1 out of 23. So now let's go back and look at that chart. And on that chart, then we were looking at the carrier frequency for cystic fibrosis. We see that for Caucasians of European descent, the carrier frequency or incidence is 1 in 23. So now let's go back to the previous slide that we looked at, and we see here that Caucasians of European descent have a 1 in 23 carrier rate. So let's look at something that you'll be doing quite often as a genetic counselor. You'll be looking to calculate the carrier frequency. You have a patient comes in who's from a specific group, and they'll be asking you, what's the probability that my baby is going to have a specific disease? Well, in the case of cystic fibrosis, if you're taking a look at two unrelated Caucasians, what's their probability? Well, the probability that both of them are carriers is 1 out of 23. And so 1 out of 23 times 1 out of 23 equals 1 out of 529. And now what's the probability that their child has cystic fibrosis? Well, even when you have two carriers, let's remember, it's only a 25% or 1 out of 4 chance that you're going to have a child who actually has the disease. So there basically are three numbers, one for the carrier for the father, one for the carrier for the mother, and then when the two of them have a child, what's the probability that they're going to have a baby that has the specific disease? So in the case of this example, if it's 1 out of 529 for the two parents times 1/4, it's going to be equal to 1 out of 2,116. So what if my brother or sister had cystic fibrosis? What's the probability if my brother or sister had CF? And my partner's family does not have any history. We are, however, both Caucasian. The probability that both are carriers is 1 out of 23 times 2/3, which equals 2 out of 69, or 1 out of 34.5. Why does she have the probability of 2/3? Well, her brother has a disease. He is homozygous for the disease, and she does not. So she is either homozygous dominant or she's a heterozygote. So go back to the Punnett square. And when two carriers of an autosomal recessive disease have a child, there's a probability of 1 out of 4 that they are homozygous dominant, 2 out of 4 chances that they are heterozygous, and 1 out of 4 that they are homozygous recessive. Since his sister does not have the disease, there are 3 possibilities left, and 2 of them are that she is a carrier, so 2/3. The answer is 1 out of 23 times 2/3 times 1/4, which equals 1 out of 138, as opposed to the previous answer from 2 people from the general population of 1 out of 2,116. In this case, we basically know that the disease is in this specific family. What if an individual with an autosomal recessive disease like cystic fibrosis would like to have a child? What is the probability of them having a child with the disease if the partner is from the general population? So in this case, it's 1 out of 23 from the general population times 1, because we know that this individual has a disease, therefore has the gene, and it's 1/2. Now notice it's not 1/4, so draw out the Punnett square, and you will see that it is 1/2. And so 1 out of 23 times 1 times 1/2 equals 1 out of 46. So if one partner is from the general Caucasian population and the other has the disease, then 1 over 23 times 1 times 1/2 equals 1 out of 46. When looking at calculating the risk for X-linked traits, different predictions of allele frequencies apply to males and females. For a female who can be homozygous recessive, homozygous dominant, or a heterozygote, the standard Hardy-Weinberg equation of p squared plus 2pq plus q squared equals 1 applies. However, in males, the allele frequency is the phenotypic frequency because a male who inherits an X-linked recessive allele exhibits it in the phenotype. So for the females, we see that it goes by the Hardy-Weinberg formula, but for males, then it's going to be p plus q equals 1. So let's look at hemophilia A, X-linked hemophilia A, where 1 in 10,000 males are born with this disease. Therefore, Q, the frequency of the H allele, equals 0.0001. Using the formula P plus Q equals 1, the frequency of the wild type allele is 0.9999. The incidence of carriers who are all female equals 2PQ, or 2 times 0.0001 times 0.9999, which equals 0.00019. That is 0.0002, or 0.02%, which equals about 1 in 5,000. The incidence of a female having hemophilia A is Q squared, or 0.0001. This is about 1 in 100 million. Since next week is Christmas and you have 1 week off and you have an exam to do, we're going to stop week 8 at this point because I think there's enough material for you, and I also know that there is nothing any further on your exam that is in part 2. Part 2 will be put up so that you can look at it during the vacation if you'd like to, but please, everybody, take some time off. Otherwise, it will be there for you at the very beginning of the last part of the semester, the next 8 weeks. So have a wonderful Christmas, complete all of your assignments, rest up, and Happy New Year and Merry Christmas, and Happy Hanukkah.